Good evening, everyone, and welcome to the IEEE EMBS Türkiye webinar. My name is Zeynep Ulaşan, and I am the EMBS Türkiye student liaison. I am happy to be your host today. Tonight, we are joined by our special guest, Professor Dr. Uygar Tazebay. He is a well-known expert in molecular biology, biotechnology, and biomedical research. He will talk about cellular liquid and liquid phase separation and its implications in human health, and share ideas that connect basic research to real healthcare solutions. Thank you all for being here with us. Please enjoy the talk and feel free to write your questions in the live chat for the question and answer session at the end. Now, let's welcome Professor Dr. Uygar Tazebay.
Okay, hello. Good evening, everybody. So today, my title is Cellular Liquid Phase Separation and its Implications in Human Health. There has been an important paradigm change in biology, in the basic sciences, and that is the involvement of liquid phase separation in cellular, in a variety of cellular phenomena, both in cytoplasm and in nucleus. So this evening, I'm going to summarize a bit this concept of liquid phase separation and its involvement in cell biology, and also a bit what we have been doing in this subject since a while. All right, so I will make an effort to explain what is liquid phase separation and how it is implied in basic biology and in human health. So this is also one of my first slides in cell biology course. This is nearly everybody's first slide in cell biology course. When we describe eukaryotic cell biology or the eukaryote cell, we mention nucleus as the organelle or the compartment that keeps the genetic material. We say, okay, the cell is surrounded by the cell membrane, and inside the cell, inside the eukaryote cell, there is cytoplasm, and there are organelles or different compartments in which particular biochemical reactions they take place. So we describe mitochondria, lysosome, endoplasmic reticulum, variety of organelles, like you all know. And when we come to cytoplasm, we don't describe much. And in general cell biology courses, cytoplasm is sometimes described as a soup in which biochemical reactions they take place, so on and so forth. So this white area or the white material is not well described, or we don't bother to describe what is happening in the cytoplasm. Whereas the cytoplasm is actually a very dynamic location, and it is not acting like a soup as most of cell biology teachers, they describe. Now, I'm going to show you a model of cytoplasm and how cytoplasmic enzymes or different biomolecular substances and biomolecular complexes, they interact with each other. This is Adrian Alcock's model, just to give you an example of how we have been seeing the cytoplasm. So in this model, Adrian Alcock have analyzed the concentration of about 250 cytoplasmic proteins, and by using bioinformatics or in silico analysis of cytoplasm, he came up with a model where these 250 cytoplasmic proteins, they are behaving together, they are interacting with each other, and so on and so forth. But this model, Adrian-Alcock model, has a problem, which is the following. The reactions are very fast, and this analysis doesn't show the reaction time or the speed of reactions. And another thing is that it is hard for the enzymes to find the right substrate in such a cytoplasmic mass. Now, as I told you, there has been a paradigm change in cell biology, and with that paradigm change involves formation of dynamic biomolecular complexes depending on the situation, both in the nucleus and in the cytoplasm. And by that, dynamic biomolecular complexes, I mean the following, which implies to a number of biomolecular interactions that take place depending on the conditions outside of the cell, that take place both in cytoplasm and in nucleus. Now, one of the best known examples is the nucleolus or nucleoli, that is a membraneless biomolecular complex. It is also said a membraneless organelle, which is inside the nucleus. And nucleolus is the site of ribosome assembly, ribosomal RNA synthesis. It is the site of the telomerase assembly. It is the site of the 7S signal recognition particle assembly. So this is the site. This is a membraneless organelle inside the nucleus, and it is basically known as the site of ribosome assembly or ribosome synthesis. Now, there is another well-known biomolecular complex, which is the stress granule, which we will be talking about in a minute. And stress granule is, again, a dynamic biomolecular complex. I say dynamic because it's not always there. It appears, or it is formed when the cell enters into a stress. And that could be starvation stress, that could be temperature stress, that could be viral stress, or that could be oxidative stress. So basically, what stress granule is doing, it is to inhibit translation by engulfing ribosomes, by engulfing active ribosomes. So basically, in response to stress, one of the first reactions of the cell is to stop translation by forming, as I said, a dynamic, gel-like, if you want, a biomolecular structure that engulfs ribosomes. There's another example, which we say a biomolecular complex, a dynamic biomolecular complex. Some says it is a physiological amyloid formation, and that is bulby antibody. And bulby antibody is, again, a biomolecular complex that is observed in woman's egg, and that keeps the egg in its potential, in its reproductive potential for years until the egg is fertilized by a sperm. So bulby antibody is one of the major cytoplasmic biomolecular complexes that keeps vertebrate egg functional for many, many years until it is fertilized. There are other granules, again, dynamic because they form and they disappear and they reform if needed, such as, spliceosome, for example, such as the PML body, such as the causal body of nucleus. And you see examples of these granular biomolecular complexes in this figure. Anyway, I don't need to recall them one by one. So now the paradigm that changed is that we don't describe cytoplasm as a soup that also contains organelles, well-known organelles such as mitochondria, endoplasmic reticulum, lysosome, trans-Golgi network, and so on, but biomolecular complexes that form and they disappear depending on cellular conditions. One of the biomolecular complexes that forms by liquid-liquid phase separation are the P granules. That is very well studied by Tony Hyman since many years. P granules are in fact granular biomolecular complexes that define which part of the C. elegans fertilized C. elegans embryo at the first cell stage is going to form the posterior part of the C. elegans of the worm, and which part is going to be the anterior part. So as you observe in this figure, the posterior part is defined by the concentration of P granules, and P granules are nearly absent at the anterior part. But interestingly, P granules, this concentration gradient is not formed by the movement of P granules to the posterior part, but rather by disappearance of P granules at the anterior part and by reappearance of these biomolecular complexes, of these membraneless organelles, if you want, at the posterior part. So they disappear from the anterior part and they reappear at the posterior part. And when Tony Hyman and his colleagues, they have analyzed the physical nature of P granules, they have observed that when two small granules, they come together, they fuse to each other like a bit like a liquid droplet like biomolecular complex, like two liquid particles when they come together. I mean, when it rains to the windshield of your car, or when it when it rains to the window of your house, you observe that these rain droplets, when they come together, when two small droplets they come together, they fuse and form a bigger droplet. So this was the observation of Tony Hyman. P granules, indeed, like droplets, they fuse to each other and they form a big one. And that was, that is how the idea that these membraneless organelles can indeed be liquid-like particles have appeared. So in fact, liquid-like particles or liquids in biology is a common subject. I mean, when you make an omelet, when you break your egg in the morning to prepare an omelet, you observe that some liquids, they come out of the egg, right? And liquids, like liquid-like particles in cytoplasm, which is considered to be a liquid. I mean, cytoplasm notoriously is considered to be a liquid environment, and having liquid particles in the liquid environment is also an observation that is common to many of us. I mean, when you invite your guests to the house, if you make a salad and you make a salad sauce, you add a balsamic vinegar inside olive oil, and balsamic vinegar inside olive oil, it doesn't mix the moment you add them to each other. Okay, so water and olive oil, if you agitate the bottle, they may look like like a mixture, but when you allow it to stand for a while, they reform. A phase separated or liquid-liquid separation is observable when you add two liquids like olive oil and water, or olive oil and balsamic vinegar to each other. So liquids that doesn't mix to each other is also a common observation. So the idea or the changing paradigm now instructs us to think about the cytoplasm like a mixture of liquids that does not mix to each other. So when we look at the cytoplasm with that eye, we can understand better how these dynamic biomolecular complexes, they form, but they don't mix to each other. Now, this is another experiment that is related with stress granule formation. Now, the experiment, the control is at the bottom, what is marked as the mock. The mock indicates a cell that is not stressed. So in the cell that is not stressed, in the red column, you can see the nucleolus. You can hardly see the cytoplasm, but nucleolus is obvious. And the moment we induce stress conditions by heat shock, by oxidative stress, or by transduction of a virus to these cells, for instance, poliovirus, then we start observing these dot-like structures. Now, the red column indicates staining of cells with a stress granule marker protein, which is TIA1, and the green column indicates staining of the stressed or control cells by mRNA dyes. So, for instance, when we transfect the cells with a virus, in this case with the poliovirus, these dot-like structures, they appear, and these are the stress granules that contain polio mRNA or that contain mRNA. Similarly, when oxidative stress is induced, again we see these dot-like structures that appear, and they are full of messenger RNA as it is marked in the green column, or when we heat these cells to 42 degrees for half an hour, or 46 degrees again for half an hour, for an hour, any temperature above 40 degrees, we observe the same thing, these dot-like structures that contain messenger RNA. So these dot-like structures that contain messenger RNA, they are not always there. That's why we call them dynamic biomolecular complexes. They are there only when the cell is stressed by virus, by oxidative stress, or by heat shock. Now, when we analyze better what are these stress granules, in this example, we see formation of a stress granule in response to dengue virus, in response to viral stress. When the cells, they are attacked by virus, a kinase, phosphokinase R or PKR is phosphorylated. In response, PKR phosphorylates eukaryotic initiation factor 2 alpha, which is involved in translation. This is translation initiation factor subunit, eukaryotic initiation factor 2 alpha. It is phosphorylated by PKR, and in response, the ribosomes, they are packaged in a gel-like biomolecular complex that we call stress granule. So these are membraneless organelles, these are granular organelles, and they are dynamic because they are formed and they disappear when the viral attack is abolished. Okay, so stress granules, they form in response to heat, in response to virus, in response to oxidative stress, and they are reversible, so they form in response to stress, and they disappear when the stress disappears. So this is one of the best studied examples of dynamic biomolecular complexes that form via liquid-liquid phase separation. Now, when we come to the liquid-liquid phase separation situation, I'm going to explain why we consider stress granules as liquid. But first, I want to make a little pause here and talk about a number of diseases that affect human health. One of these dramatic diseases is the ALS disease or amyotrophic lateral sclerosis, the disease that Stephen Hawking had survived for many, many years. And we know Stephen Hawking has a mobility problem. He was always in the wheelchair, especially after the mid ages. And the condition of Stephen Hawking was a motor neuron disease that is called ALS. And the problem here, the motor neurons are lost little by little, and at the end, the person loses the ability to move. Now, there are a number of familial ALS mutations, and one of that is TDP43, another one is FUS, another one is HNRMP A2 family. They are affecting RNA metabolism, splicing, transcription, but interestingly, they affect stress granule formation. So FUS, for example, is a stress granule protein. It is important for stress granules, the dynamic formation and disappearance of stress granule, as well as TDP43, which is again involved in stress granule formation and disappearance. Now, another group of familial ALS mutants, they are mapped in VCP, for instance, that has to do with autophagosome formation, optineurin, sequestosome 1, or UBQLN2, which has to do with autophagosome with degradation of these biomolecular complexes. So either increase in the formation of these complexes, such as stress granules, or decrease in disappearance of these complexes or degradation of these complexes when they are not needed. These two conditions, they are associated with familial ALS disease. Now, a third group of familial genetic mutations, they map to cytoskeleton-related functions, tubulin functions, profilin functions, NF or P150 mutations. So, but what I find interesting is that formation of stress granules, when it is affected, they cause familial ALS. Now, again, studies of Tony Hyman group, but mainly one study that comes from his group, the first author is Avinash Patel. This is published about 10 years ago, but this is, again, an important publication. I have analyzed what happens when FUS is mutated, like in the variants of familial ALS gene mutation cases, whereas the wild-type FUS. Now, these authors, they have purified familial ALS variants of FUS, such as FUS glycine 156 glutamate. And when the FUS protein is purified and incubated in the buffer conditions, at the beginning, wild-type and familial FUS mutation have behaved the same. They form these droplet-like biomolecular complexes, but the more they are incubated at the room temperature, for instance, 4 hours of incubation, they are looking again similar, but in the longer incubation times, such as 8-hour incubation, the mutant FUS have lost the droplet-like characteristics, and it became rather solid-like biomolecular complex. Now, this was an important observation because it means wild-type stress granule protein has a droplet-like or liquid-like characteristic, whereas the mutant protein loses the liquid-like characteristic and it becomes rather a solid-like biomolecular complex. Now, in the single plane illumination microscopy, you can observe this liquid-like characteristic again, two smaller droplet. They have this round liquid-like or droplet-like structure, but when the two comes together, they fuse and they become a bigger, again, droplet-like structure, which is reminiscent of liquid structures. And in the electron microscopy, the mutant FUS, mutant FUS, that the variant is the cause of familial ALS or neuromotor disease, has rather a solid-like structure. Now, this was the first observation of Avinash Patel in 2015, published in Cell, and that was one of the major observations. Now, when we analyze different types of neurological diseases, such as frontotemporal degeneration, ALS, Parkinson disease, or Alzheimer's disease, we observe a similar phenomenon. I mean, if after the death of these diseased people, the brain sections are analyzed, we observe pathological aggregates in all cases, like the tau pathology in the Alzheimer, or the ALS pathology, or the TDP43 or FUS pathology. We see this aggregation in the pathological aggregates. When these proteins are expressed or overexpressed in cells, notoriously a phase-separated liquid-like, puncta-like biomolecular complexes are observed. And when these proteins are purified and they are observed in buffer conditions, they look like droplet, round, liquid-like particles. So then the paradigm shift was the following. Perhaps the pathological aggregates, they don't happen at once. These are biomolecular condensates that should remain like liquid, but when they lose the liquid characteristics and when they solidify, they form these pathological aggregates that are the causative agents in those diseases. And interestingly, all of these are neurological disorders. So the idea here is that these pathological aggregates, indeed, they are in the soluble state, but when they lose the solubility and they form aggregates, they cause pathological conditions. Whereas, like in the example of bulby antibody, in fact, when these monomers, they come together in an arranged state, they can be called physiological amyloids, and they have a physiological function. Again, like in the case of bulby antibody, the physiological function is to inhibit the metabolism of the cell so that the cell can be kept alive or can be kept with its biological potential for decades. So, in fact, there are two sides of the coin. These aggregates, when they are ordered and when they are reversible, they do not harm or they don't cause disease. Rather, they are physiological aggregates, like in the situation of nucleolus, like in the situation of stress granules, different types of bodies, like in the situation of spliceosome, like in the situation of bulby antibody. These are necessary membraneless organelles that keep the cells functional. But when they go out of control, when this aggregation is irreversible, and that leads to pathological situations like in the multiprotein pathologies, like in the ALS disease, Alzheimer's disease, or frontotemporal dementia. So what we've been doing on that, in fact, we have not established a hypothesis in our laboratory to analyze membraneless organisms, but rather we started from the other side of the coin. We analyzed genomes. We found a gene that is conserved in all eukaryotes, and we wanted to understand what it does. This is, for instance, the encoded protein from this gene. The name of this gene is coil domain containing 124 or CCDC124. This gene is conserved in all eukaryotes. Here there are some examples. Aspergillus nidulans is the mold, is the bread mold, or is the mold that grows on your bathroom wall. C. elegans is the worm. Zebrafish is the example of a vertebrate model, and human, we put human as an example of conservation. So this is a conserved gene. It is conserved also in plants. For instance, in Arabidopsis we have it, in alveolites we have it, in excavates we have it, in all eukaryotic organisms. Now, when we analyze better the biophysics of this protein, here you see charge distribution. This is positive charges versus negative charges. They alternated. This protein has a large disordered domain. Sorry, it's here. This is order disorder distribution. It is mostly disordered. We have a region that seems to be ordered, and that region maps to the RNA binding domain of this gene. Again, a disordered region, and again, an RNA binding, putative RNA binding domain of the protein, and the rest is disordered. So this was charged protein and alternating charge, ordered and disordered domains, mostly disordered, and so it was fitting to the biophysical characteristics of a protein that is involved in liquid-liquid phase separation, or biomolecular complexes that form without a membrane. So we analyzed this protein. First of all, we have shown that it is truly an RNA binding protein. This is a student in our lab, Özge Arslan. She's now a postdoctoral fellow in Finland. She studied, so she has shown that the protein binds RNA. This is an RNA binding protein, and besides, it is upregulated in a variety of cancers. Now, this upregulation in cancers, it happens in multiple biomarkers of cancer, but the RNA binding characteristic is what I was focusing on since that time. Now, that protein is found, that protein, coil domain containing 124, is found in another membraneless biomolecular complex, and that is called small extracellular vesicles. In small extracellular vesicles of aggressive tumor cells, there are only a couple of proteins, and one of the, they have found only 6 proteins, and one of these 6 proteins, this is the group of Susanna Godinho in London, they found CCDC in this membraneless secreted biomolecular, if you want, biomolecular complex. So one of the explanations of why CCDC is in cancers may be because it triggers metastatic characteristics of cancer. But what I find interesting is that, again, CCDC is in a membraneless biomolecular complex, such as small extracellular vesicles. Now, another group that have analyzed CCDC or its yeast ortholog LSO2 was the group of Wendy Gilbert, from the Broad Institute of MIT, and they have shown that CCDC is in the ribosome, and it regulates restart of translation after stress has abolished. So during stress, ribosomes, they hibernate, ribosomes are packaged in stress granules, and after stress, these stress granule packaged ribosomes, they open up and they start translation, and one of the regulators of this phenomenon is CCDC. Now, other groups, they have mapped position of CCDC. Basically, it was Roland Beckman group. They mapped position of CCDC. You see CCDC here in red inside the ribosome. And again, after hibernation, or once stress is over, for restarting of translation, CCDC was functional, and it is here. This is the position of CCDC. And again, Roland Beckman's group have shown that, in fact, when the virus enters inside the host cell, and in the example of SARS-CoV-2 or the COVID virus, when it infects host cells, basically COVID virus interacts with a subset of ribosomes in the host cell, and this subset of ribosomes that help COVID virus translation is the subset of ribosomes that contain CCDC. So basically, these groups, they have shown the involvement of CCDC in stress granule functionality, and more importantly, in the involvement of stress granule in SARS-CoV-2 interacting viruses. So also, our analysis have indicated that CCDC was involved in membraneless organelles, such as centrosome. In this case, here you see centrosome by the marker protein gamma tubulin. So these dots, they indicate centrosome position, and the green dots indicate CCDC position. So CCDC was present in centrosomes, which was one of the earliest known membraneless organelles. And CCDC was located in the cytokinesis in dividing cells at another membraneless organelle, which is the midbody. So midbody is less known. It is here. So when two cells, they divide, like you see in this figure, these are cytokinetic cells, they divide. And here in this round structure, you see midbody. So when we analyze in dividing cells, like in this situation, here you see 1, 2, 3, 4 dividing cells. In the synchronized division, you see CCDC 1 to 4 in the middle of these cells, in the midbody, in all these 4 cytokinetic cell cases. So we analyzed cell division when we knock down by CRISPR the CCDC 1 to 4. We observe cells, huge cells, multinuclear cells with cytokinesis defect. And now we also analyzed presence of or interaction of CCDC with a nucleolus or nucleolar protein, which is nucleophosmin. First, we have shown that in the nucleoli, CCDC exists, and in the nucleoli, CCDC interacts with nucleophosmin. So when we stain nucleoli, this was the work of Gamze Çakırca in our laboratory. When we stain nucleoli with anti-nucleophosmin antibody, you see these dots. And when we stain the cells with CCDC antibodies, we observe the same dots, so they colocalize. Also, nucleoli is observable by bright field microscopy. So we have shown that nucleophosmin and CCDC, they interact. And in fact, we have modeled, this is thanks to Ömer Güllülü's work together with Gamze Çakırca. We have modeled interaction of nucleophosmin and CCDC inside the nucleus. This is when nucleophosmin is wild type, and this is when nucleophosmin is mutated. So CCDC interacts with mutant nucleophosmin also, but the interaction is different. Or, in our model, these two proteins structurally, they don't interact with the same way, if you want. So we have analyzed the nucleophosmin CCDC interaction, and we have also found other common interactions to nucleophosmin and CCDC, and most of them was ribosomal proteins. And on top of that, we have carried out biotinylation, bio-ID analysis, or in other words, we have marked CCDC with biotin ligase, and therefore biotin ligase marked CCDC whenever it went inside the cytoplasm or inside the nucleus. It biotinylated the proteins that are in close proximity. It biotinylated all proteins that are at 7 nanometer proximity to CCDC, and therefore we have carried out a high throughput analysis by mass spectrometry, high throughput analysis of all proximity partners of CCDC 1 to 4, and this is done by Merve Tuzlakoğlu Öztürk in my laboratory, and this have indicated large number of RNA processing proteins, some cell division and cytoskeleton components, and viral life cycle primary film assembly or stress granule proteins that was close proximity to CCDC. For instance, again, Merve Tuzlakoğlu and Ömer Güllülü have shown unequivocally that Stau factors that are for stress granule reversal were interacting with CCDC. So now what we are proposing is that CCDC have a role, especially in stress granule function, and possibly one of the roles of CCDC 1 to 4 is reversible action of stress granule. In other words, dissolution of stress granules once stress have bypassed. So we have also shown that CCDC is in stress granules like TIA or G3BP labeled cells. You see stress granule formation. When we stain the cells with CCDC, we stain the same locations, meaning that G3BP and CCDC are colocalized, or CCDC is a stress granule partner protein. Now, in fact, I'm going to pass all that. This was what I wanted to tell you at this time. So one of the main paradigm shifts in biology is the presence of liquid-liquid phase-separated dynamic biomolecular complexes in the cytoplasm and in the nucleus. And when we come to our research, we have proved that CCDC is involved in the formation of at least stress granule and perhaps nucleoli formation, two membraneless organelles. But we don't know whether CCDC phase separate on its own or with the presence of an RNA protein or an RNA macromolecule inside the nucleus or inside the cell. We have not so far analyzed a time-lapse movement or translocation of CCDC 1 to 4 in the cell. Again, CCDC seems to have different phosphorylation, putative phosphorylation sites, and we don't know whether post-translational modification or phosphorylation or citrullination, if you want, of CCDC is involved in its functionality in these membraneless organelles. Now we believe CCDC have a function to reverse phase-separated organelles like reverse stress granule formation, or rather it helps dissolution of stress granules, but we have not formally proven the role of CCDC in these stress granule biology. And also we have not proven, I mean, we have some evidence indicating that CCDC is a moderate causative agent in some neurological pathologies, but we have not proven, again, unequivocally, the involvement of CCDC in neurological pathologies. So with that, I want to end my presentation. I want to thank especially Merve Tuzlakoğlu Öztürk, which was together with me in the lab in the initial stages of CCDC biology. Ömer Güllülü, Sinem Gül, Özge Arslan, Gamze Çakırcaine, Kübra Hacıbeyoğlu have done a lot of work of CCDC biology, all wonderful postdocs now. Hilal Civelek, a PhD student now in Basel, analyzed CCDC involvement in plant biology together with Yelda Özden Çiftçi, which is a PI at Gebze Technical University Molecular Biology Department. We have been generously supported by TÜBİTAK, Turkish Ministry of Development, and EMBO short-term fellowship program, as well as Fevzi Akkaya Scientific Research Funds and European Science Foundation, which have supported mobility in my laboratory. So with that, I want to end my presentation. I hope I've been able to give you a sense of importance of liquid-liquid phase separation, in biology. I can have any questions you may have right now. Thank you for listening to me.
Thank you very much, Professor, for this great presentation. Now we will move on to the question and answer session. If you have any question, please write them in the comment section. Okay.
Maybe I can go to YouTube and... No, it will be a mess if I go. I'd better not.
Okay. Let's start with our first question from Zehra Mercan. What is the relation between stress granule dynamics and FUS mislocalization in ALS patients?
Yeah, FUS seems to be one of the major components. It is a causative agent in ALS disease. So it's an important protein. It is a stress granule protein. The stress granule dynamics is as follows. In the ALS patients, stress granule seems to lose liquid-like characteristics, and it has a tendency to rather gellify or solidify. So the relationship between stress granule dynamics and FUS mislocalization is that when FUS mislocalization happens, then stress granules, they lose their biophysical characteristics, and they have a tendency, or FUS has a tendency, to form a pathological aggregate which causes, which abolish normal cytoplasmic functionalities and derive the cells to pathological situation, which is in this case ALS disease. So there's a strong relationship, very strong relationship between stress granule dynamics and FUS mislocalization.
Yes, our next questions from Sude. How might SARS-CoV-2 NSP1 exploit ribosomal interactions to interfere with CCDC1-2 force functions?
Yes, very good question. Very good question. Now, this battle, I mean, even though we have survived this pandemics 2-3 years ago, okay, or 5 years ago, let's say, this battle between virus and the host cell is, is very long, is million years old. So viruses and host cells, they have existed on this planet for millions of years, and they know each other very well. So we know SARS virus only very little because we have started to study this virus 2-3 years ago. But the cell and the virus, they know each other since millions of years. So, in fact, when the virus infects the cell and the immunity response and the cell's response, or the cellular immunity or intrinsic immunity's response, is to gellify the cytoplasm, by the formation of stress granules. But the virus has also some weapons, and the virus tries to liquidify back to the initial situation. So there is a battle between virus and host cell. The host cell tries to gellify the cytoplasm or tries to form the stress granule, and the virus tries to dismantle, open it up, and liquidify. So now, I don't have an answer to your question, okay? We don't know perfectly what is happening, but we have a sense, and we believe, or I believe, what is happening is the following. The host cell tries to, entangle the virus by solidifying the cytoplasm, and the virus tries to open it up. So the functionality of this interaction, what NSP1 is doing, NSP1 is trying to reconvert cytoplasmic conditions back to suitable conditions for the virus. So it interferes. So again, I don't have an answer to your question, but it may interfere with what is inhibiting itself to translate. So maybe one hint is that CCDC is changing the ribosome or the ribosomal structures such that it is not suitable for the virus to translate, and NSP1 attacks specifically to CCDC containing ribosomes to convert them back to translation competent state for the virus. That is as much as I can answer you. But we are very much interested with your question, and this is exactly the question in my lab, the question that we don't have a clear answer so far.
Thank you. Next question from Tuana. Could you discuss any recent progress in small molecules or peptides that can modulate FUS fuzz behavior?
Exactly. So this not only for FUS. So as I told you, there is a paradigm change, and this paradigm change is that a number of neurological diseases, and perhaps more than that, many diseases, they have to do with formation of aggregates, formation of biomolecular complexes that instead of staying liquid, they gellify or solidify. So one of the answers to that was, what if we design peptides by using artificial intelligence, perhaps, or by using bioinformatics? What if we design peptides that target these mostly disordered domains of aggregation-prone proteins, and that would inhibit their aggregation? So this is a wonderful question, in fact, and that is discussed in the field. There are large number of groups that are working on discovering new peptides that are going to bind to disordered domains and that are going to keep these, entangled disordered proteins or these aggregation-prone proteins And once these peptides, they interact with these aggregation-prone proteins, these proteins won't aggregate anymore, like in the situation that we see in Parkinson's disease, Alzheimer's disease, ALS disease, or dementia, or other diseases. I mean, there is myositis that affect muscles. There is a number of so-called multi-system proteinopathies. For instance, Paget disease of the bone is a bone disease, but it is the causative agent is again aggregation of these disordered proteins. So not only neurological diseases, but there are a number of diseases that when the aggregation is abolished by peptides, perhaps these diseases may be avoided. So wonderful questions. Recent progress in small molecules or peptides, I know mostly peptides that have helped the aggregation to dissolve in the experimental conditions. I don't know any clinical study that have addressed that have used peptides, but so far in the basic sciences, peptides may affect or inhibit aggregation of these pathological variants.
Thank you, sir. Our next questions from Hatice Beyza Kayacan. How do liquid fast separation dysfunctions contribute to the pathogenesis of disease such as neurodegenerations or cancer?
Okay, so how is a good question. I'm going to summarize with what I have learned from what I have read. So liquid phase separation. So basically, these biomolecular complexes, they have to be in the liquid form because, as I have discussed in my talk, they have to be reversible. So they have to come together, form a liquid-like biomolecular complex, like in the case of stress granule. But once the stress is over, they have to open up and disappear or dissolve. Now, liquid phase separation, in fact, most of the proteins that are in these phase-separated biomolecular complexes, they have large disordered domains. Disordered domain means a domain of the protein that can have multiple conformations, multiple structural conformations. That is why we call them disorder. They don't have an order or they don't have a well-established structure, but rather they have different conformations. So, and that gives their liquidity. Okay, so they have different conformations, so-called, they have multivalent interactions. So they have different conformations, they interact with multiple RNA or protein, but there is this multivalency, so they interact with multiple macromolecules, and besides they go from one conformation to another one. So this gives them like a liquid, like you see, I try to show you with my dancing. So they are like liquid. But the mutations, the causative mutations, they map important and particular positions of these disordered proteins. And with these mutations, the proteins are not disordered anymore, but they have a tendency to have a certain well-established order. Or by time, with the mutations in other, they were multivalent, okay? So they interact with multiple RNA and protein with multiple factors. And by time, these variants, they start to get a tendency to remain rather like a gel or to solidify. So how do liquid phase separation dysfunction contribute to the pathogenesis? By losing this liquid-like structure, by losing their disorganized state or disordered state and having an established or pre-established ordered state. The moment they have like I have shown you in my talk, an agglomeration prone order, then we start observing the disease because they are not reversible anymore. They are not liquid anymore. They come together with liquid-liquid phase separation, but they have a tendency to remain solid or to solidify, and that is the start of the problem. So the cytoplasm and nucleus, they have to remain liquid-like for long periods of times or for decades for the biology to function.
Thank you, sir. Our next questions from Samet Başkaya. Are there any structural insight from cryo-EM or other methods that suggest how to disrupt pathogenic NSP1 CCDC 124 interactions without harming normal ribosome recovery?
Yeah, thank you. Samet, this is a very good question. One thing I have to explain is that so far we couldn't see NSP1 CCDC direct interaction. Now, NSP1 interacts with ribosomes that contain CCDC. But I don't think it interacts directly with CCDC. Okay, so this is an important point. If NSP1 interacts with CCDC 124, which is not our observation, then it could be interesting to find small molecules that might block, for instance, NSP1 CCDC1 interaction. But however, this is not our observation. So NSP1 interacts with CCDC 124 containing ribosomes, but not directly with CCDC. So we don't have any structural insights about the nature of this interaction. I mean, also about the nature of NSP1 ribosome interaction.
Okay, thank you, sir. Our next question comes from Milos Popic, which in vitro or in vivo models have been most informative for studying FUS-related phase separation defects?
Well, in vivo models, first of all, now there is this possibility that one could get cells from the patients and convert them to any cell type by induced pluripotency, IPS, and then analyze it. So one of the most instructive in vitro models are the cells that are obtained from patients and that are converted back to neuronal cells, and then they're, in patients with familial ALS disease, now you can obtain their cells and study FUS-related phase separation defects directly in the patient cells. So this was a wonderful model because you can understand what is exactly happening in the patient cells. And the other informative, well, of course, I mean, you can take FUS and transfect any cell type, HeLa cells, and so on. I don't want to talk about these studies, okay? So one can have causative FUS mutants, study them in HeLa cells. This is also interesting, but up to a certain point. I think one of the most interesting models were patient cells by induced pluripotency are converted to neuronal cells, and there the FUS-related phase separation defects are studied. Another group of in vivo models were FUS-defective mice, obviously. So you can, by CRISPR, introduce causative familial ALS mutants to vertebrates, to mice, and there study what is happening. So the most informative ones are patient cells converted to neuronal cells by induced pluripotency, not only for FUS, but also for other, TDP43, HNRMP family, namely A1, SOD1 mutants, there are SOD1, I mean, there are different types of ALS models. And to answer your question, one of the brightest in vivo models were transgenic animals, and in vitro cell line models were cells derived from the patients and converted back to differentiated cell types.
Okay. Thank you for your explanation and presentation, sir. And
all wonderful questions. Thank you, everybody, for asking these impressive questions. They were all very, very good.
We can wait a bit in case maybe there are any other questions.
Yeah, of course.
Okay. There is no questions. Okay. Thank you all for joining us today and for being part of this webinar. We hope you enjoyed the session and learned something valuable. Before we end, we would like to invite you our upcoming big events, EMBCon. Okay. It will bring together experts, students, and professionals to explore the latest in biomedical engineering and health technologies with inspiring talks, workshops, and networking opportunities. You can find more details and register on our website. Okay. Once again, thank you for your time, and we look forward to seeing in your future events.
Thank you.
And yes, also thank you, sir.
It was a pleasure. Thank you.